Paterson. I'm currently an employee of Atomic Energy of Canada Limited, as well as I'm a graduate student at McMaster University, and I'm currently studying the effects of neutron irradiation on DNA. Both ACL and McMaster University are members of a core group of labs in Canada that, in the event of a nuclear accident or nuclear emergency, so a dirty bomb situation or situations where sources have been distributed or released into the general public by accident, these labs are able to take blood samples from the people involved, look at the chromosome aberrations, the DNA aberrations, and determine whether that person has received a dose that would be medically concerning. If that is the case, at that point we pass that information off to a doctor, and those people are then treated accordingly. In the case of an accidental exposure to the public, we would receive blood samples. We take these blood samples and we culture them in our lab facilities, which just means growing them in a really nutritive environment. And because blood samples don't normally divide, we have to stimulate them to progress to metaphase. This is the phase of the cell cycle where we can best see the chromosomes and by default best see chromosome damage. At that point, we put the cells onto slides, stain them for a good contrast, and we're able to look at them under our microscopes. So we're looking for chromosome aberrations, which is an indication that a radiation dose may have been received. When we come across chromosome aberrations under the microscope, we mark down the aberration, and over a spectrum of 100 cells, the number of aberrations, we can correlate that to a dose received based on previous experiments undertaken within the labs. The more chromosome aberrations we see in each person's sample correlates with the dose received. So more aberrations means higher dose, fewer aberrations, lower dose. This is a biological safety cabinet, and when we're doing cell work, we want to make sure that we minimize any bacteria, viruses, other pathogens that might get into our tubes with our cells. Because we're putting the cells in ultra-nutritive media, there's also the potential for unwanted pathogens to grow in that media as well. So the biological safety cabinet is a sterile area where the air that comes in is HEPA filtered, the inside is thoroughly decontaminated, so we can be sure that when we open our tubes with our cells in them, that no external pathogens or bacteria are getting into the tubes, and this way we know that the results we're getting are actually the correct results. If we had bacteria growth in our samples, suddenly that skews our results and makes the work that we're doing invalid. My work changes on a daily basis. The scope of aberrations that we see on a daily basis is different. The different types of radiation that we use to mimic accident situations changes all the time. Sometimes we use gamma, sometimes we're doing stuff with X-ray radiation, other times we've just started to work with neutron radiations. And so it's just really neat to constantly evolve that line of work. I really think that the work I'm doing here and the work that other groups are doing in the radiation biology field is very important and will benefit the health and well-being of Canadians.